my name is Hiba Hussain, and I'm a fourth-year biology student at Northeastern participating in the CaNCURE co-op program. And today I'm going to be telling you a little bit about my project and what I've learned so far from this amazing experience. So my project is entitled Screening High-Risk Patients for Multiple Myeloma Precursors via the PROMISE study. And I have had the pleasure of working in the Gabriel lab at the Dana-Farber Cancer Institute. So what is multiple myeloma? Multiple myeloma is a slow-growing hematologic malignancy. It is a cancer of the plasma cells, so the cells in your body that produce antibodies. They're antibody factories, and they help fight off infections. But when too many of these plasma cells start being produced, and the ratio goes from healthy cells to malignant cells, this can progress into multiple myeloma. So there's many stages of the disease, and these are precursor conditions for when there's a low ratio to plasma to malignant cells. These are termed as MGUS, or monoclonal gammopathy of undetermined significance, and smoldering multiple myeloma, or SMM. However, when this, as I was saying, when these become, this ratio becomes too much, it progresses into multiple myeloma. And the symptoms that characterize multiple myeloma are hypercalcemia, kidney failure, anemia, and bone lesions, all signs of end-stage organ damage. And currently, the problem with multiple myeloma diagnosis is that it's often seen as a watch and wait type of condition where patients usually get to this end-stage organ damage before interventions are taken. But the Gabriel lab is working to change that by identifying these precursors and screening patients for them and informing them about this so that intervention can be taken at an earlier stage. And a biomarker for multiple myeloma is known as an M protein or M spike. So when these plasma cells start to overproduce, this can be identified based on the type of clone of the antibody, and that can be identified on an SPEP or serum protein electrophoresis. So the PROMISE study in particular aims to screen high-risk populations to prevent multiple myeloma before it even begins. So who exactly do we mean by high risk? Well, Black and African American individuals are twice as likely to have multiple myeloma. First-degree relatives with multiple myeloma are also high risk, as well as those with chromosomal mutations, as the ones that I have listed, specifically the ones that I have listed on the slide here. The current method of detection for multiple myeloma involves SPEP, or serum protein electrophoresis. It's very advantageous because it's cost-effective, it's efficient, it's the standard in pathology labs because it involves taking the proteins from a patient's blood and running it through a gel. However, it's disadvantageous in that it's very subject to human bias because it's qualitative, you're looking at fluorescence, and it's imprecise for those with precursor conditions because sometimes the ratio of the plasma cells that are healthy and normal to the malignant ones could be very low because there's just so few cells. So this is where mass spec is an amazing tool. It's advantageous because it's highly sensitive, it's precise, and there's minimal human error involved. However, it is costly and it's still undergoing validation to be used in clinics. This is a little bit about my workflow, my day-to-day activities in the lab. So the providers, the physicians will identify high-risk patients in the clinic or through the PROMISE survey that is sent out on the website. Blood samples are collected from these patients and the serum or plasma is isolated for the liquid handler. This machine prepares the samples for the MALDI-TOF mass spectrometer, so it puts them on this MALDI plate. We run the MALDI plates on the MALDI-TOF mass spec, and this provides us with one data point. And then we run the blood sample, the serum or plasma, on the opsolite protein analyzer, and this provides us with another data point. So we take those two data points together to assign a diagnosis and inform patients within 4 to 6 weeks. So this co-op has really taught me a lot, and I'm very excited to see what the Gabriel lab does next. So they hope that the system will start to be used in clinics with the mass spec and the opsolite in 1 to 2 years after undergoing FDA validation. And the end goal of the PROMISE study in particular is to screen 50,000 patients for precursors, which is a very significant number. As far as technical skills, I really gained familiarity with the MS workflow, mass spec workflow that I mentioned on the previous slide, and I developed an understanding of the assay that uses magnetic nanoparticles. So that's that connection to nanomedicine that we see in the field. Personally, I found a lot of motivation to go digging for the answers to my questions about the field, about the newest research in the literature itself, and that really confirmed my interest in oncology as I hope to become a physician scientist in an academic setting in the future. And of course, this experience could not have been possible without Dr. Gobriel and the mass spec team, the CaNCURE team, and the NCI grant. It could not have been possible. Thank you so much.